In Chapter 9, we talk about the second type of cellular reproduction, meiosis. Meiosis serves two primary functions. Let's go ahead and make it clear that meiosis is only involved in sexual reproduction. This is in contrast to mitosis, which is involved, of course, as we pointed out in Chapter 8, in growth and repair and asexual reproduction. Meiosis is only involved in sexual reproduction. Meiosis does two things. One thing it does is it reduces the overall chromosome number. Each organism has a specified number of chromosomes. In the case of humans, that's 46. And so you can see from this diagram from our text that it's necessary in the process of reproduction to split the chromosome number in half. So each individual donates half the number of chromosomes to the offspring. This means the egg has 23, the sperm have 23, and when they come back together, then the zygote, which is going to develop into the offspring, has the complete number of 46. We also want to make sure we're familiar with the terms diploid and haploid. If we look carefully at this diagram, we notice that diploid is represented by the abbreviation 2N. And this 2N equals 46 means that the diploid number of chromosomes in the case of humans is 46. Haploid is represented by the letter N, lowercase n. And so in this case, N, the haploid number of chromosomes, equals 23. In the case of humans, and all other vertebrates and most other animals, the adult organism is diploid, or even the juvenile organism, but the organism itself is diploid, and the gametes are haploid. So the eggs and the sperm are haploid, and the organism itself is diploid. We also want to make sure that we're familiar with the concept of homologous chromosomes as we approach this chapter. All chromosomes, in the case of humans, are arranged into homologous pairs. What we mean by this is the following. A homologous pair of chromosomes contains the same genes but different alleles. Alleles are different. So if we look down at this part of our slide, we see that alleles are different forms of the same gene. So we can see this clearly if we look at this example on the screen. Here we have a pair of homologous chromosomes. Here we have an allele that's located on those chromosomes. In this case, the allele is a gene that codes for flower color. On the bottom chromosome, the allele codes for white flowers. On the top chromosome, the allele codes for purple flowers. So in other words, the homologous chromosomes have the same genes, but different versions of those genes. Think about it this way. In the case of a human, you could build a human being, at least in the case of females having two X chromosomes, with just 23 chromosomes. But humans have 46. This means there is redundancy. There are two copies of every single gene that's found in the body. At least again, as I'll make this point again, in the case of females, because females have two copies of all the genes found in their X chromosomes. Males, in contrast, have a different X and Y chromosome, so they only have one pair of all the genes found on the X chromosome and one pair of all the genes, or one copy rather, of all the genes found on the Y chromosome. Now you know what sister chromatids are, but we review it here just to be sure. Sister chromatids are what is produced when a chromosome replicates itself. So if we look at our screen, here we see a chromosome in a non-duplicated form, and here we see a chromosome after it has duplicated itself. The key thing to remember is that sister chromatids are not the same as homologous chromosomes, because sister chromatids are held together at their structure referred to as a centromere, and sister chromatids are identical. So unless a mutation occurs, sister chromatids are going to be exactly the same, 100% the same. So a homologous pair of chromosomes is going to exist during part of its life as each chromosome will exist as two sister chromatids held together at the centromere. Here we have what's called a karyotype. The karyotype shows us all the chromosomes that are found in a human being. So humans have 2 pairs of each chromosome. They're numbered based on length. This is 2 pairs of 1, 2 pairs of 2, 2 pairs of 3, and so forth, all the way down the line. When we get to the sex chromosomes, we see that this is a female because this particular individual has 2 X chromosomes. If it were a male, then this individual would have 1 X chromosome and 1 Y chromosome. So the sex chromosomes do act a little bit differently. As I pointed out previously, in the case of males, we don't have 2 copies of every gene that's located on the sex chromosome, just 1. Females, although, do have 2 copies of even the genes located on the sex chromosomes. So to review, humans are diploid organisms because we have 2 pairs of every chromosome that's located in our body, with the exception of these sex chromosomes. Genes located on the X and Y chromosomes, as we've pointed out several times now, are not duplicated in males, but they are duplicated in females. Females are XX, in other words, they have 2 X chromosomes. Males, of course, are XY. Now we get to the idea of meiosis, and the basic idea of meiosis is depicted on the screen. What we see is that it's similar to what we learned in the case of mitosis, except we are splitting the chromosome pairs in half. And so that's the big difference. If we look here at meiosis 1, what we notice is that the homologous pairs of chromosomes are located along this region of the cell known as the metaphase plate. So I'm pointing to the metaphase plate right now. Now, it's an imaginary line, there's not anything there physically, but it's the plane along the middle of the cell at which the chromosomes line up. This separates the chromosomes. It takes the cell from diploid to haploid. Another difference in meiosis from mitosis is that we have 2 steps to the process. Meiosis 1, in which we separate the chromosome pairs, and meiosis 2, in which we separate the sister chromatids. So now in meiosis 2, we have 2 cells, and these cells are haploid. I can tell these cells are haploid because this is a homologous pair, if you follow the pointer. I can tell it's a homologous pair because these chromosomes are identical in length. We notice here in meiosis 2 that we only have 1 of this pair found in this cell and 1 of this pair. And the same thing applies to the other pair of chromosomes. Therefore, we can tell that these chromosomes are haploid, not diploid. We undergo a division in meiosis 2 in which we split the sister chromatids apart, and we end up with 1, 2, 3, 4 cells when all of this is said and done, and all of those cells are haploid. The n equals 2. Remember, we started out with 4 chromosomes, and so the haploid number of chromosomes is 2. The n equals 2 means haploid number of chromosomes equals 2 in this particular case. In the second half of our chapter lecture, we'll talk about what happens in each of these stages. But for now, we want to point out a couple of the key differences. To review, meiosis has 2 divisions, and meiosis reduces the chromosome number from diploid to haploid, cutting the chromosome number in half overall. In meiosis 1, we're diploid, and the chromosomes in each homologous pair line up along the metaphase plate, this imaginary line, and in meiosis 2, the sister chromatids themselves separate.